Hi, I'm Hiroshi Yamakawa. I'll give you presentation entitled Whole Brain Reference Architecture to Evaluate Biological Plausibility of Human-like Artificial Intelligence. As background, why biological plausibility should be assessed. First, brain-inspired software is significant in 3 points. High affinity for people, a promising way to build an artificial general intelligence, and vessels for mind uploading. Second, the advances in neuroscience and deep learning made to build brain-inspired software. Then, we would like to ask, is biological to lower the barriers to entry for such software development? Our non-profit organization promote to develop a brain-inspired artificial general intelligence by mimicking the architecture of the entire brain. This approach is based on the central whole brain architecture hypothesis shown below. The brain combines modules, each of which can be modeled with a machine learning algorithm, to attain its functionalities, so that combining machine learning modules in the way the brain does enables us to construct a generally intelligent machine with human-level or superhuman cognitive capabilities. The feasibility of the whole brain architecture approach continues to increase. Firstly, because the advances in neuroscience made the brain a subject that can be mimicked. Secondly, because the advance in deep learning made it possible to process information like human brain. When can we complete AGI by referencing the brain? It's essentially the timing that we can find an architecture that enables human-like intelligence. Generally speaking, the computational cost of learning is far greater than its execution. And the computational cost of searching for the architecture that organisms have found through evolution is even greater. Human intelligence is realized in the 86 billion neurons that make up the brain. Upper limit of search space is capped by a connection combination of these neurons, if we ignore further detail. The brain-inspired AI approach greatly narrows the scope of search by adding brain constraints to the architecture. Then, within the limited scope, we search for architectural solutions that exhibit human-like capabilities as AGI. Then, brain-inspired AGI will be completed shortly after the ability to search for AGI architectural solutions exceed the size of the design space constrained by the whole brain reference architecture. The amount of papers and data in the field of neuroscience is vast. Through them, it is not easy to estimate the degree to which the software implementing the algorithms is consistent with the truth of neuroscience. How can we overcome this difficulty? Importantly, the range to which brain-inspired software should mimic is not all of the neuroscience. Getting a general picture of the brain is essential to recreating its intellectual mechanisms, because the brain is composed of a tightly coordinated network. Nevertheless, the scope to be grasped can be narrowed down, and even that scope need not be completely exhaustive. Then which aspect of the brain should be focus? It is specifically a mesoscopic level of brain architecture. Because, mesoscopic brain architecture clearly contribute to intelligent processing. As a matter of fact, functional mappings between artificial neural networks and natural neural circuit are claimed in mesoscopic level. For example, the visual cortex is mapped to convolutional neural networks. More generally, minimum granularity to be referenced is moderate. Too coarse a granularity results in insufficient constraints from the brain. Nevertheless, referring to too much detail will complicate the description and increase the computational complexity of the model calculation. So, the bridge between algorithms and neuroscience should be built on a mesoscopic brain architecture. We defined a brain reference architecture as a static reference model that describes mesoscopic level architecture of the brain. It consists of circuits and connections between circuits. As function hypotheses, each circuit has annotation of function and each connection has annotation of meanings of signals. We have defined the minimum description unit called a uniform circuit. Each of uniform circuit is neural population that transmit a computationally identical kind of signal. we have defined the brain information flow, BIF, as a specification format to describe a whole brain reference architecture. Please refer to the poster presentation by Mai Sasaki in this symposium, for detail. Brain-inspired software can be described by component diagrams. And the descriptions of the BIF and that of component diagrams are almost same. Therefore, we can evaluate the biological plausibility by comparing the component diagrams reflecting the software with the BIF that summarizes the neuroscience findings. Because both of component diagrams and BIF diagram represent the flow of information, we can compare them. In a word, circuits on the BIF can be mapped to components on the component diagram and connections on a BIF can be mapped to a port on a component diagram. So, we can assess fidelity value, that is the degree to which the software associated with the component diagram reflects the BIF. Then fidelity value could be defined based on a similarity of optimal aligned BIF and component diagram. Then what kind of processing can calculate fidelity value? The step 1 in the evaluation process is to determine the optimal alignment between the BIF and the component diagram by performing a comprehensive set of 3 comparisons. First, the semantic identities of functionality on circuit and function on component are compared. Second, the semantic identity of the function on connection is compared to the meanings of the signals on the ports. Thirdly, the mapping between the BIF and the component diagrams as a graph structure is evaluated. The remaining differences in the optimal alignment thus obtained represent a lack of fidelity. In step 2, the fidelity value can be calculated by using some function that transforms the amount of difference. We investigated method for assessing the biological plausibility of brain-inspired software. This method uses the whole brain reference architecture, which described the mesoscopic level architecture of the brain. In the future, evaluation methods should be refined to be quantitative and eventually automate. Various issues remain to be addressed as follows. Dealing with diversity for component diagrams and for BIF diagrams. Get involved neural activity phenomena for evaluation. Consideration of anomalous systems for evaluation. Evaluate the validity of the BIF description itself. The whole brain reference architecture aggregate architectural information at the mesoscopic level of the brain. And this reference architecture plays a central role in the development of brain-inspired AGI. This is because our whole brain architecture approach, in short, aims integrating machine learning component like the brain. In this presentation, we have described how reference architectures are used to assess the biological relevance of implemented software. However, this reference architecture can also be used to guide the development of a system for a specific task. The reference architecture can also be guide brain-like merge of components developed in different projects. This reference architecture is manufactured as the form of a brain information flow. First, the anatomical structure and physiological phenomena are collected from papers and data. In addition, possible hypotheses about the computational function are also collected, but if these are insufficient, we attempt to hypothesize a function that fits the structure. We call this process of hypothesis building the SCID method. Furthermore, we have a database for sharing/integrating the accumulated knowledge. The whole brain architecture approach is steadily progressing toward the realization of brain-inspired AGI, by using this development scheme. Thank you for watching.